genomics of zoonosis. It's a very fantastic topic. And today he's here to present us summary of a lot of things he's been doing, doing research on this viruses and the genomics of the viruses in Latin America specifically. So I'm going to give him the room, and thank you for being here with us.
Thank you, Rebecca. Okay. So I hope everyone can hear me and see my slides. If not, just let us know through messages or things like that. Okay, so first of all, I would like to thank you very much for this opportunity, to Professor Rebecca Sanchez for organizing everything and for bringing me here to Costa Rica. It's important to say it's my first time in Costa Rica and my first time here in the University of Costa Rica. I'm really happy to be able to come here and participate in these training activities and also strengthening collaboration within Latin America. That's something I really believe in. We can do much better and much more things together, working in a collaborative way. So as Rebecca kindly introduced me, I am Gabriel Wallau. I'm a public health researcher. And within Cruz and BNITM, my work is mostly focused on arboviruses, arthropod-borne viruses. But today I'm going to talk to you about genomic insights on zoonotic viruses spread dynamics in Latin America, with some examples of arboviruses and some other examples of other zoonotic viruses I have been working with in the last years. So the topics I will address in my talk today is the high impact zoonotic viruses, the case of arboviruses and influenza virus. Well, there are a lot of things to talk about influenza virus, but I'm going to tell you a little bit more about the experience I have been collaborating with some people from Uruguay in detecting and trying to reveal the spread of this virus in South America. With these examples, I'm going to talk about current surveillance and knowledge gaps, most focused on Brazil, where I have more experience, but also South America and Latin America. Examples of genomic surveillance of zoonotic virus in Latin America, and the opportunities and challenges that emerge from it, focusing on bioinformatics. So, talking a little bit more and give you a background about zoonotic viruses. Zoonotic viruses, as we all know, are viruses that infect animals, including humans. Of course, we have a view of protecting humans from being infected by these viruses, but humans is only an additional species that these viruses are able to infect, right? So basically, zoonotic viruses, we can find a lot of zoonotic viruses in wild animals. All vertebrates host a range of zoonotic viruses, domestic animals, and also vector-borne transmitted viruses. So this is the main view about the hosts and which host those viruses infect. And as we know as well, those viruses induce huge problems to humans regarding health, regarding economic loss, and so on. We recently had experience with the COVID-19 pandemic, which is caused by a zoonotic virus that has as main reservoir bats, but that spill over to different species and end up infecting humans and spread in humans very successfully. Oops. So talking a little bit more about arboviruses, arbovirus, as the name says, arthropod-borne viruses, are zoonotic viruses that are transmitted by arthropods. And those arthropods, although this picture here and many publications only focus on mosquitoes, in fact, there are several kinds of arthropods that transmit those viruses, like mites and even flies. But of course, mosquitoes are more widespread around the globe, and they are more associated with the human populations. That's why a lot of the efforts to characterize arthropod-borne viruses are focused on mosquitoes. So regarding arboviruses, we have three main cycles: a epizootic cycle, which the mosquito transmit the virus among domestic animals, and eventually this infected mosquito feed on humans, transmitting the virus to humans. Here in this case, humans are dead-end hosts, so they do not contribute to further transmission, but of course they suffer the consequences of the infection. We also have an enzootic cycle, which are mainly maintained among reservoirs, wild reservoirs. And the same thing happens. The wild sylvatic mosquito species feed on those hosts, maintaining the cycle in the wild. But eventually, when the humans came in, and the mosquito feeds on human and transmitting the virus to humans. Usually, again, this kind of transmission ends up where humans are dead-end hosts. There is no further transmission. But eventually, some of these viruses here, like chikungunya and dengue, they do establish a urban transmission cycle, like we have in the right part of the slide. So in the urban transmission cycle, we can have the urban epidemic cycle, like we know very well for chikungunya and dengue virus, where there is no need for an extra reservoir species from wild animals or domestic animals. And the transmission is maintained successfully among the human population and the mosquito vector, right? Every time I talk about zoonotic viruses, and particularly on arboviruses, I always bring this slide here, showing the impact, the distribution of those viruses and the impact they have on human populations. And this map is very good because it highlights several of those viruses. We know very well dengue, which worldwide distributed, mostly in the tropical region. But this is a map that's also outdated because it's not showing the spread of dengue virus to the subtropical and temperate regions in both hemispheres of the globe. We can also see here chikungunya virus, which is also another arbovirus worldwide distributed in the tropical region, which more recently was also detected in subtropical and temperate regions. Yellow fever in Africa and in Brazil, and some other more obscure and neglected virus like Mayaro and Oropushi virus. Another thing that this outdated map shows us is that Zika virus, for example, we know the Zika virus circulation continues to circulate in Latin America, but in fact, there are clearly current community-acquired transmission of Zika virus in Southeast Asia, sustained transmission in Southeast Asia. So it's really a static map that should be updated with more recent information, but provides you the whole view about the impact of those largely distributed viruses across the globe. So here is one of the articles I developed within my team and in collaboration with other researchers, only using public data. So at the time of the Zika epidemic, around 2015-16, we decided to reveal all the evidence regarding the transmission of the main arboviruses, dengue, chikungunya, and Zika, and which our main question here was to address which were the main paths that the viruses went through to arrive in the Americas and then to spread to South America. And what we saw here with this review, and where we included new analysis, of course, is that different lineages of dengue from different serotypes, chikungunya and Zika, they usually arrive through the Caribbean islands, where they amplify substantially. And because of movements of tourism and so on between the countries, then the virus eventually get a ride and then arrive in different countries, both in South America and North America, and of course, in Central America. So I'd like you to keep in mind this pattern here, because I'm going to talk to you later about some new findings we are having from different lineages and different viruses, which are continuously corroborating this key path to enter the Americas and spread within the Americas. So moving on with the background, I would like to talk a little bit more about arbovirus in Brazil. So while surveillance of arbovirus in Brazil is relatively good. So we do have good data since 1986, where we started to detect outbreaks of dengue virus across Brazil. But in fact, the first cases of dengue-like symptoms, because at that time there were no molecular diagnostics, was dated back to 1800s in Rio de Janeiro. But here in this graph, we have a more recent picture from 2001 to 2019, showing that in Brazil, we have this pattern of wave-like cases occurring year after year, right? And you can see the numbers of dengue cases are very large. But I'm gonna give you, I wanna give you this information because I'm gonna show you what happened this year regarding the number of cases, which was really explosive, right? Another thing I'd like to highlight here in terms of dengue cases in Brazil or arbovirus case is that this red part of the graph showing the Zika cases that were first detected in 2015, 16. And at the same time, in the same time frame, we also detected chikungunya virus. So at the time, we had the triple epidemic, dengue, Zika, and chikungunya, happening at the same time and spreading across the country, right? And after this 15, 16 triple epidemic, we had a drop in the number of cases in terms of dengue and other arboviruses. But since 2019, we start to detect a huge number of dengue cases again, and it's continuously increased throughout the years. Here is a map of Brazil showing the main regions of the country, and you can see that dengue cases occur in all regions of Brazil, right? So it's endemic to the country since the 1986, from what we have registered, but we can see that we have a lot of cases throughout Brazil. So, and why do we have it? Because, of course, Aedes aegypti is distributed all over the country. We can see here a very old map from 2007, and the presence of Aedes aegypti in almost all municipalities of the country, except by the Western Amazon and the southern part of the country. And why we detected that? Here we have the Aedes aegypti distribution and the number of cases in those regions. And there is a clear barrier here in the southern region and in the Amazon region. And it happened because in Brazil, although Brazil is known to be a tropical country, we also have a subtropical region, right? In the southern Brazil, we have a subtropical region when the temperatures, here we have the temperatures throughout the year in this map here. And yellowish colors are average temperature, which are lower during the winter time. And because of this temperature here, there were a climatic barrier not allowing Aedes aegypti to spread to the southern part of the country, right? And in the Amazon region, the explanation is that this is the Amazon forest. So urban centers were very small there, do not sustaining continued transmission of dengue virus. So, but what happened throughout the years? During 2021-22, this paper here, some colleagues from Cruz studied and found out that what was occurring was a fast expansion of dengue virus, also crossing these barriers. You can see there in the Amazon region, we detected now a lot of dengue cases, even in those municipalities within the Amazon forest. And you can see here in the southern part of Brazil, there is almost no barrier now for dengue cases. Now we know that Aedes aegypti is also distributed in the southern cone of Brazil and South America, and also all over the Amazon region. So there is a clear eroding of the barriers, the climatic barriers that existed in the past. And most of the cases have been detected now in these regions where the population is completely naive. There is no immunological barrier. There is plenty of vector mosquitoes able to infect and transmit the virus. And then we have the perfect situation for massive outbreaks in those regions, right? I did not bring here some slides about Uruguay and Argentina, but the same thing is happening there. There are quite huge outbreaks, record of cases year after year in Argentina, Uruguay, and Paraguay. And in Brazil, here is the last data from this year. We are topping 6.5 million cases this year, right? So this is the new record of cases. We are topping, we surpassed 4,000 deaths up to now, and there are a lot of deaths still on investigation to see if it was caused by dengue or not. So that's the main picture from 2021-22, and I'm gonna update you with some new findings from our research regarding the dengue lineages. But before that, do you remember I told you about chikungunya virus that arrived together with Zika in Brazil? Few people talk about chikungunya, right? Of course, Zika was a huge problem because of the consequences to babies and mothers and so on. But chikungunya is an arthropod-borne virus that's transmitted by Aedes aegypti and cause long-lasting symptoms. People stay with symptoms for months and even years. So it's very detrimental to the population. And in Brazil, genomic surveillance captured this virus entrance in the country in the same way we captured for Zika virus. We detected two main lineages, two main genotypes. One arrived from the Caribbean islands to the north of Brazil, the genotype Asian Caribbean genotype, and one arriving directly from Africa, entering here at the Bahia state, the ECSA genotype. And at that time, these authors here evaluated the potential spread of the virus throughout the country. And of course, because the vector is there, both lineages could spread countrywide. The curious situation now for chikungunya is that this lineage here, the Asian Caribbean, almost disappeared. For some reason, we still don't know why. But the ECSA genotype really took over and spread countrywide, also causing outbreaks in Paraguay, Argentina, and Uruguay. And to finish this background information, I'm going to talk a little bit more about other less known arboviruses. Here, it's a group of arboviruses, the Venezuelan equinocephalate virus and the West equinocephalate virus. That also happens in South America, in Latin America as a whole. And you're going to link with this information with some recent findings from Uruguay and southern Brazil, where we characterize a recent outbreak of West equinocephalate virus. Okay, so with this, let's enter and discuss a little bit about some of the findings we are generating using genomic surveillance. So here's one recent paper where we characterize this spread of the chikungunya virus to the southernmost state of Brazil. Remember, chikungunya in this region was not frequent before. So before 2001, we never detected more than 52 cases per year. And all those cases, or most of the cases, they were imported from some endemic regions of Brazil. There were track record of traveling of people coming from those regions, from southeast, from northeast, where chikungunya is endemic. But here in this paper, we characterize the largest outbreak with 220 cases, diagnosed cases. Of course, there are many more that are asymptomatic, but we characterize 220 cases of chikungunya in the southernmost state of Brazil. It happens in the northeast part of the state. We sequenced 23 genomes. All of them belong to the CSA genotype I told you about just briefly before. We detected that this virus was transmitted from some endemic region of Brazil, probably Rio de Janeiro or São Paulo. But now what we found in this paper is that the virus was introduced from one endemic region, but now it established community transmission, right? So we can see here several genomes that clustered together, showing that this virus was establishing community transmission through the competent vectors that are in the region. And what we are investigating now is that, well, in the next year, do we need to have other introduction from endemic region, or this virus here could remain during the winter period and then increase in frequency in the next year, establishing them endemically in the region. So here I'm going to talk a little bit more about dengue virus. So I showed you before that dengue is endemic in Brazil. The 4 serotypes already circulated in the country. But in the last years, the last 10 years or 15 years, we only detected dengue 1, serotype 1, and serotype 2, right? Serotype 3 was one serotype that disappeared within the country for a very long period. But using genomic surveillance and a large collaborative effort with few Cruz colleagues, but not only, also with CDC Puerto Rico and some other institutions across the region, we managed to detect very early on the re-emergence of dengue virus serotype 3 in the country. So we detected the virus emerging in the country, and we characterized it emerged probably from the Caribbean islands, right? Again, reinforcing this pattern. We published that in the Emerging Infectious Disease, and why characterizing this re-emergence of the virus was important, because as we know, for dengue virus, when the population gets infected with one serotype and then get infected with a second serotype, then we can have very serious consequences of more hemorrhagic dengue fever and more severe consequences to the world population. So that was a really big, important warning we raised here. And I didn't have time to bring to you the latest data here, but we are seeing that it was not a single introduction that that died out. In fact, we are detecting that dengue 3 is really spreading throughout the country, and that's really concerning regarding what's going to happen in the next years. And here is another paper we published with some colleagues regarding the multi-introduction and countrywide spread of dengue 2, genotype 2, also called the genotype cosmopolitan in Brazil. So here we generate 237 genomes. We collected all the information available, and we detected that at least 4 introductions happened from Peru to Brazil, right? And that genotype 2 is now spreading across the country. Here again, the latest data is showing that genotype 2, within the dengue 2 serotype in Brazil, we already detected 2 different genotypes. Genotype 2 had very low frequency within the country. But now we are detecting that dengue 2 genotype 2 cosmopolitan is taking over, is replacing the previous genotype. And now several groups are investigating why it's happening. Is it more effective in replicating in the mosquito body? Is it more effective in infecting the human population, inducing high viremia, and maybe having different symptoms in terms of dengue fever? So that's another point where we are focusing on within Brazil and trying to collaborate more with neighboring countries to understand the spread patterns and what are the consequences in terms of public health. Yeah, here this is another picture from this paper showing that in red you can see dengue serotype 1, and in blue serotype 2. You can see that serotype 3 and 4, there's almost nothing across the country, only this new detection of dengue serotype 3 in some states. And you can see that there is a mix of patterns. We have the different Brazilian states here. Some states we have dengue serotype 1 as the most prevalent, and for some other states like PE, Pernambuco here, or SE, the dengue 2 is the most prevalent. But the thing is, with this data, this is the data coming from the national surveillance system, and there is no genomic information here. We cannot really extract the information, which is the genotype. We only know the serotype with this information here. That's why adding the genomic information over it can give us more detailed information about which genotype are circulating and if we do have this replacement occurring in the country, and what's the meaning of that. So why these examples? Of course, I already told you briefly that when we do have following infections with different serotypes, maybe we can end up with antibody-dependent enhancement, which may induce more severe dengue cases, and it's really important to map that and follow it. We also have some other interesting thing to analyze now that we are able to monitor using genomic surveillance in very much detail regarding the antibody waning. So we still do not have surveillance in terms of immunological surveillance of the population, but we know that for any kind of virus, but particularly for dengue virus, when a person gets infected, then antibody is produced to neutralize the virus. But then with time, there is an antibody waning. The amount of antibody decreases over time, right? So if it does happen, what it means in terms of public health measures? Should we vaccinate again now that we do have some vaccines available for dengue at some point in time here? And during this antibody waning phase, when is the particular time point where we can have, for example, antibody-dependent enhancement and things like that? And here's a recent paper showing that after large epidemics, most of the population have higher antibody responses. It then takes about 1 to 3 years for the population to become sufficiently susceptible, right? So that helps us with this information coming from the immunological surveillance project, help us to understand the cycles of epidemics, right? What's the timing? Why sometimes the case drops for 1, 2 years and then it rises again? This is a very, very important information that we are looking for with some collaborators, but that the information is very scattered within Brazil and also within the region. Well, another initiative that I was mainly involved as conceptualizing it and helping to deploy it was regarding the urgent need for global surveillance of arboviruses. So after the COVID-19 pandemic, we established across the globe the infrastructure to generate genome of pathogens, high-impact pathogens. But although dengue is a high-impact pathogen, in fact, the whole globe, right? The whole population is vulnerable. Up to now, there was no initiative to really generate genomic surveillance data and share it in a timely manner. So we proposed together with GISAID to, we wrote this letter published in the Lancet Global Health, proposing to expand genomic surveillance for a urgent global need. And then we established a database within GISAID for arboviruses, for now focusing on dengue, chikungunya, and Zika virus, to allow the researchers to not only generate the data, but share it timely with other researchers. You notice in these last slides how important it is to share, to generate this data and share this data to be able to perform aggregated analysis to really detect the introduction and the transmission of the virus to different countries, understanding the situation within each country, and then trying to come up with insights regarding the further spread and what are the consequences. But if we do not share the data, then our conclusions or insights will be very limited. So that's why we proposed this where 54 countries, researchers from 54 countries joined this initiative to this call to generate more data on dengue and other arboviruses and to share it timely to perform some aggregated analysis. Here's the EpiArbo. It's a database within GISAID You probably interacted in the past with EpiCough and maybe EpiFlu for the ones that work with influenza virus. So now we have EpiArbo. People can share the data, can perform even within EpiArbo some preliminary analysis, seeing where your new sequences clustered with other sequences from each country and so on. And we can also characterize some mutations using Bank server and track which are the countries or regions that are submitting the data in real time. And here you can see the serotype frequency and the genotype frequency. Some of the things I showed to you in papers before. Now you can see it in real time without publications, without a delay in terms of reviews and so on. So we can immediately see your results in an aggregate analysis here. And one important thing to emphasize is that for those that already work with KeySaid protects the data submitted or the data generators. That builds trust that people can share the results and nobody will just pick up the results and do whatever they want, right? So that's a way, I think, and I share this vision with KeySaid, that's one of the most effective way to share the data and to generate some real-time or almost real-time insights that will have direct consequences in terms of public health. So, and to finish this part about arboviruses and, well, these most well-known arboviruses, I'm going to talk about the other less known arbovirus the next slide. I always bring this figure from this paper. I think it's really, really nice paper where the authors try to evaluate the impact of global warming and the changes in the environment and the climate in terms of transmission of arthropod-borne viruses, right? Not only arthropod-borne viruses, they also analyze malaria, but I'm going to talk about arthropod-borne viruses. And what they saw here you have in the x-axis the number of months where transmission is, if you go to the right side, to the reddish side of the spectrum, you have an expansion of the transmission season. So the temperature will be better for the mosquito to breed, to develop, and to transmit the virus. And if you go to the other spectrum, then the transmission season would be shortened, right? And focusing on Latin America, you can see that most of the region are in this part of the spectrum. So as global warming goes on, right, in this situation, business as usual, without any kind of intervention, then we're going to have more and more enlarged season, transmission season, that will end up with more human cases, inevitably. So let's talk a little bit more about other neglected arboviruses, the Urupuchi virus. Maybe you have heard about this in the last years. There are some news about this in Science and Nature and some other sources. We are, as a team, collaborative team within the Cruz Genomic Network, we are helping to characterize this little known virus as called by science. So the Urupuchi virus is another arthropod-borne virus, but it's not mainly transmitted by mosquito. It's transmitted, the main vector is biting midges of a specific species in Brazil. And this virus here is has a segmented genome of 3 segments. Here are the segments that generate different proteins that are important for the virus replication cycle. Here are the biting midges. We have here the main transmission cycle. Mostly this arbovirus, it's neglected because it mostly, the transmission cycle mostly happens in the sylvatic environment, in the Amazon region, right? So there are some mosquito species and also biting midges, which is not here in this scheme, that get infected and infect birds, but also sloths and some other South America non-human primates and rodents. And eventually, those species came and feed on humans, and humans fell ill with dengue-like symptoms, right? But what's happening in the last year, since 2022, is that we detected may be related with these climate changes, that also is changing the environment in the Amazon region. We detected a huge population growth of biting midges in the Amazon region at the first place, right? And associated with that, we started to detect a huge amount of human cases in the region. And that's where we started to work on these virus. Here in 2023, in fact, 2022, 23, because this period of the year where the transmission occurs is during the end of the year, 2023, and turning of the year to 2022, 23. But here you can see in 2024 what happens. A virus that was mostly restricted to the Brazilian Amazon region now spread countrywide. And interesting enough, we start to detect communitary transmission in those other regions, because reviewing all the data, the main biting midges, which is the main species, which is the vector of Oropouche virus, is also widespread distributed in the country. So here is the finding. We recently made available a preprint. We submitted for review now, but the genomes are available since several months. We characterize the increase in cases in the Brazilian Amazon region. Here we have most of the Brazilian states from that region: Amazonas, Roraima, Rondônia, and Amapá. And here is a very interesting picture. Here, for example, during January to June 2023, we detected the first cases of Oropouche in the human population. But during the turn of the year, from 22 to 24, we detected a huge amount of cases. And you can see here, Oropouche case accounted for most of the human cases, showing dengue-like fever, right? So very few cases were left for dengue and some other arbovirus that circulate in the region. We also detect that this Oropouche virus is a new reassortant lineage, right? Of course, the data from 2010 backwards is very scarce. There are very few genomic data, so it's hard to really ascertain and to evaluate those hypotheses. But with the data we had, we characterize a new reassortant of segments that came from different lineages of Oropouche virus circulating in the Amazon as a whole, in the Amazon from Peru and from other neighboring countries. And here is a more recent study we are conducting to understand this spread from the Amazon to other parts of the country. We are seeing here, we have Brazil there with the main biomes. And here, the different parts of Brazil, we are seeing that there is an increase in cases in the southeast part of Brazil, where São Paulo and Rio de Janeiro is, and also in the northeast part of the country. And this virus really established communitary transmission, because in those regions, the number of cases do not stop to rise. There is more and more cases every week. So here's the analysis we are performing with 33 genomes from different regions. We can see two main clades here. Some of the introductions came from this clade from the Amazon state, and some other from the Acre state. But it's clear that there is communitary transmission, a cluster of cases showing this in different states. So in the last example, with VEV, West equinocephalite virus. VEV, again, is a virus that is maintained in a sylvatic cycle among birds and mosquitoes. But eventually, when there is El Nino and some climatic changes, the population of the vector increased substantially, and then this vector feeds on equines and humans. Most of equines and humans are dead-end hosts. They do not contribute to transmission, but they suffer with the virus infection, even with encephalitis and death. So we are characterizing this West equinocephalite virus in South Brazil, Uruguay, and Argentina. Here's the first paper where we detected the West equinocephalite virus in equines in these 3 municipalities in red in this map of the southernmost state of Brazil. It was accepted in emerging infectious disease, and we characterized that it was a lineage that was circulating there for a long, long time, right? It was not a new introduction from other countries. It was circulating there, and probably because it is maintaining a sylvatic cycle, surveillance do not reach those pockets, and we had no information about this virus, the circulation of this virus. Here there is an error, sorry about that, but this is another paper where we characterize all the West equinocephalite virus cases in equines in Uruguay. Here are the cases and the spread of cases throughout the country. It happened very, very rapidly because, as I showed to you, the main reservoir are birds, and birds travel very rapidly across the country, spreading the virus, and then impacting directly the equines in the region. So here are the new genomes we generated. We call it a nucleate in South America. Here there are some genomes that were available by other groups, but most of these genomes were generated with this study here. And here with this clade where we zoom in, we detected 3 main clades, but these 3 main clades were, they spread very rapidly. So we can see subclade 1, subclade 3, and subclade 2. They really spread all over Uruguay in a matter of few weeks. So it's a submitted paper. And the last example regarding viruses, let's talk a little bit about influenza virus. So influenza, I won't go into detail, but just to show you the complexity of influenza virus. So influenza is a respiratory virus, a virus with a segmented genome, and it's well known to infect a range of animals, right? It's a very versatile virus because it has these segmented genomes. A lot of reassortment happens, generating new lineages that are more fit to infect some species or another. But what is happening regarding influenza in these last months, for our perspective in South America, but in fact in the last years? So there is active spread of avian influenza virus, mainly that lineage, HPAI 2344B. This is an HPAIV virus. It's a high pathogenicity avian influenza virus. And this virus was first detected in 2016, but the current outbreak started in October 2020, in the middle of the pandemic. So that's why we didn't heard a lot about this. We were only hearing about COVID-19 at the time. But in October 2020, it started to infect and impact poultry production in Europe. And then this virus spilled back and spilled over between birds of poultry production and wild birds. And because of migratory paths of wild birds, this virus started to spread. And then it spread to North America. And then from North America, it entered South America and it started to spread in the Pacific coast and then went to the Atlantic coast. But what I'm going to show is that we started to detect some unusual patterns. So here, just to give an idea about the impact of avian influenza virus in wild animals. This is a picture from an island in UK, showing the number of birds in that island. And this is the year 2022, after this virus almost erased that population from the island. So this virus caused mass mortality in animals, right? But what we started to detect when it arrived in South America, not only South America, but particularly in South America, we started to detect more consistently that not only birds were suffering the direct consequences of avian influenza virus infection, but we started to detect mass deaths of mammals, right? We have seals here and some big group of pinnipeds have been affected. I don't know if you remember, in Peru, there were these first cases of mass mortalities in seals. It came out in the media. People were really concerned because we have an avian influenza that now jumped to a mammal. Maybe it will adapt to mammals and start spreading within mammals. And that's just one step before transferring or spilling over to humans and maybe causing a new pandemic. Here is a graph, an internet graph showing the number of human cases caused by this specific lineage. So it has been detected since a long time ago. There are a lot of human cases, but all those human cases were sporadic with no further transmission. And here is the current, the most recent years, the number of human cases. So human cases continue to happen as this virus spread through the wild animals populations. In Uruguay, so where we added information with our colleagues from Uruguay. So they started to detect this mass mortalities in seals and also in birds. And they were collecting those samples in wild birds from the coast and seals from the coast, but also in poultry farms from within the country. And what we wanted to test here is that, well, is the same virus that is infecting the poultry within the country and the seals and this more coastline spread of the virus? So when we sequenced several strains of the virus and we performed some phylogenetic analysis, we concluded that no, it's a different lineage. So here you can see in pink the first sequences from poultry production in Uruguay. And here up there, you can see the lineages from this outbreak in seals and birds, seabirds, right? Another thing that was quite surprising here is that these two infection waves had two different time frames. So this happened at the beginning of 2023, and this happens months after the middle of 2023. And there is really a decouplement, those two transmission of those two lineages were decoupled in time, showing that it's really a different transmission pattern that's happening within the country and in the coast part of the country. And more concerning than that, we found that all those sequences, all those lineages from pinnipeds, including a human sequence detected in Chile, contained two specific mutations, this Q591K and D701N, which allows, because the influenza virus, we do have a lot of information about the influenza virus, we know specifically which mutations allows the virus to adapt and replicate more efficiently in mammal cells. So these two mutations occurred in the RNA polymerase of the virus, and they are known to replicate and adapt the virus for mammal adaptations, right? So we can see here in the left phylogeny, we performed some ancestral reconstruction, and you can see that most of the sequences come from pinnipeds and some birds, but the birds are scattered within the pinniped clade, suggesting now that the transmission occurred within pinnipeds, from pinniped to pinniped, and then eventually a bird gets infected from the seals, right? And this is another paper we also are characterizing with our Uruguayan colleagues, where we are showing a specific case of transmission of influenza virus in coatis, another mammal, right? Which are very much abundant in the region. But there is only this report up to now. It's still under review, but we characterize that those coatis were probably infected from wild birds that, visit those same places. And that's another example of mammal transmission, mammal to mammal transmission of this specific lineage. Just to conclude here, I don't know if you are aware about H5N1 transmission in USA. in cows in USA. It's highly concerning. That's the same virus, only a different lineage, right? It has a different set of segments, but that's the same virus from the same lineage that now is spreading from cow to cow, and eventually we have more than 10 cases now in humans, if I'm not wrong, in the US. It's really, really alarming. So in the last part, I'm talking too much here. The last part, I just would like to pinpoint some of the developments required regarding tools and bioinformatic tools for biogenomic analysis. Some of the things I did within my group and also in collaboration. So at the beginning of the COVID-19 pandemic, we come up with this idea to have a genomic surveillance engine, right? Where we could enter sequencing reads from any kind of pathogen, and then it will run a set of core functionalities, and then we would be able to generate reports, have data security in place, and share this information very rapidly. It's important to say that, well, this idea was conceptualized during the COVID-19 pandemic, but in fact, it was a little bit before that, during the Zika epidemic. I started to work on what's called now viral flow back in 2015-16. And then it was growing and growing, and we come up with this idea during the COVID-19 pandemic. That was the original idea. So we managed to develop some of this whole idea. One of the things we managed to develop was viral flow. Viral flow is a pipeline, is a workflow that does exactly what I mentioned to you before, but only for viruses. So we perform very rapidly genome assembly of viral genomes, and we can run several core analyses to generate standardized outputs that can be used for reports or for publications. So here's the whole idea of viral flow. We have a public code. It's in GitHub. We always kept in mind while developing viral flow to have a workflow that works both in personal computers and multi-user servers, large computational clusters. We implemented that with the latest technologies using containers and container environment. And here I won't get into detail, but those are all the steps that are performed by viral flow in a systematic way. You can pass one sample or 1,000 sample, and it will run the same steps for everyone and compile the outputs in a nice manner to use it afterwards. This is the new version of viral flow, what we call viral flow 1.0, where we expanded it. During the COVID-19 pandemic, we launched viral flow 0.6 only for SARS-CoV-2 virus, but now we have 1.0 where we expanded its functionalities. So as long as you have a reference genome with a GeneBank ID, you can pass this GeneBank ID to viral flow. It will collect the genome, readapt the code, and run some core agnostic models, right? Of course, some very specific analysis, the name is saying, will not be run. For example, pangolin is specific for SARS-CoV-2. Nextclade, you can run Nextclade for influenza or SARS-CoV-2, but it will not run for some other viruses. And another thing we worked on regarding viral flow for this new version is that we wanted to be able to share the reads, but only the reads that came from the pathogens we are investigating. Because even if we employ different methodologies to remove the host reads from our sequencing reads, we may end up still with some sequences from the human genome and from other microorganisms from that specific sample. So we develop specific functions where we can write down the specific reads that map it against the pathogen, and then you can easily share those reads without ethical concerns. Here's an implementation of viral flow 1.0. It generates a coverage depth map, which are iterative. You can zoom in, zoom out, and see the regions that are well covered and regions that are not well covered. and use this to adapt your wet lab protocols to be able to get better genomes in the next run. Yeah, this is a nice graph. My student did it, extracting the data from only Brazilian data, right? Generated from Brazilian institutions. We wanted to see how frequent viral flow was used during the pandemic. And it was really reassuring to see that viral flow was surpassed even some commercially available pipelines, because people are really using, they think it's useful in the genomic surveillance pipeline. And we also developed what we call viral genome DB. This is still under development. It's not open for everyone, but this is something that will address some of the things we wanted to address with the whole idea of the genomic engine. So viral genome DB is a DB from database, but in fact it's a system that includes a database. And this database is fed with the metadata from the samples and the genomic data, right? So we can pass as input to viral genome DB the output of viral flow, and then we can plot all the information within this viral genome DB. We can see the main lineages of a single run, or an entire month or an entire year. We can see the genome coverage, if it's good, if it's not, in different kinds of plots. Viral genome DB also automatized the process of submitting the data to different databases. Now, currently, it's working for GISAID, so we can submit consensus sequences from different viruses, SARS-CoV-2, different arboviruses, and we are working for influenza now. And one further development is that we will allow to submit reads directly. And of course, the user can also download all the data. So we can download all the intermediate files generated by viral flow if you want to check in more details the results. Oh, yeah. And viral genome DB also generate these reports here. Those reports are the main way of communications, at least within Brazil, with the Ministry of Health and at different states and municipality levels. So every time we generate a report with the lineages and lineages of concern of different pathogens, we generate with one click this report and send it directly to the stakeholders and people that take the actions to mitigate the spread of the virus. So it's already automatized, and now we are working as many groups across the world. We are working on what we call Deep Virome Explorer to have a very consistent workflow for viral discovery, for discovering the pathogen X or these new pathogens, viral pathogens that are highly divergent from the one. Messages, arbovirus burden in Brazil, Latin America is increasing due to the circulation of new lineages and linked to travel and also globalization, travel, and climate change. Recently, increasing incidence is more pronounced in subtropical regions. So there is a clear pattern of spread of viruses in those regions where the population is naive and it causing huge outbreaks. Surveillance of others on the pathogen are possible and provide key information for non-pharmacological and pharmacological intervention. I gave you several examples here. However, it should be linked with timely genomic surveillance. We need to be able to share this data in a safe way to connect this data with data from other countries and be able to perform aggregated analysis. But of course, all of this is only possible through robust and easy deployable bioinformatic tools, because now the bottleneck is not generating data anymore. The bottleneck is analyzing data. So we need to be able to analyze the data in a consistent way, in a rapid way, to share this critical information and take actions based on it. And with this, I'd like to thank you. Of course, all of this work was not done by me and would not be possible only with my working hours. It's something based on a huge team. This is my team particularly. but this is only part of a large network that includes the Fiocruz genomic network, Rede Genômica Fiocruz, and collaboration with several institutions from within Brazil, from other countries, that we established during the COVID-19 pandemic. With this, thank you, obrigado, graças.